In the last video, we talked about how membranes are selectively permeable, meaning they only let some things across. In this video, we're going to talk about how it is that they actually move material across the membrane.
Talk about that. There are two types of membrane transport. The first type of membrane transport is called passive transport. So passive transport is when the cell can move material in and out of the cell membrane without putting any energy into it. In fact, passive transport can continue after the cells are dead. Active transport, the cell must actively move the things across. That is a transport that requires a cell to do something or to use energy. So material moves because of a phenomenon known as Brownian motion. You may have noticed that sometimes if you have light streaming in your window, just the right light, you can see little specks of things floating in the air. There's tons of stuff floating in the air, but you can sometimes see them, and when you're floating, it looks like they're just literally kind of bouncing all over the place. And the reason why is that the flecks of things you can see, maybe dust or whatever it is, are hitting things that you can't see, like oxygen molecules and nitrogen molecules and so forth. And that motion is what we refer to as Brownian motion. So everything that has energy, which is everything, there's nothing on Earth that doesn't have some kinetic energy, otherwise they'd be at absolute zero, temperature-wise. But everything that has energy is vibrating. And the hotter it is, the faster it vibrates. But no matter where it is on Earth, it's got some kind of energy. And so those particles, everything is vibrating. And when the big things that we can see, and sometimes can't see, when they hit each other, it appears to move randomly. It's kind of like, I've got a quick video I'd like to show you.
Smoke from a straw is injected into a small container. A lid keeps it in place. It looks like tiny jittering points of light. When a straw burns, the smoke produced is mostly bits of carbon which haven't combusted. Under a microscope, they catch the light and appear to glimmer. But why do they randomly dance around? The movement of the smoke is caused by collisions with invisible air particles. As the air particles move about, they knock the smoke first this way, then that.
Let's take a minute and review. Brownian motion causes a phenomenon known as passive transport. Specifically, all passive transport is a type of diffusion. Diffusion is the random movement of molecules from an area of higher concentration to an area of lower concentration. That's a lot of words to basically say that they spread out. So let's define some of those things. So first off, we say that diffusion occurs down a concentration gradient, or from high to low, but we kind of need to know what a concentration gradient is. So first off, concentration is how much material there is in a space. So something that is very, very sugary, for example, like a lemonade that's very, very sugary, is highly concentrated in sugar. Gradient is a difference in two things, and you're probably familiar with this most from working with tools like Google Docs and Google Drawing and slides and things like that. And that is when you want to have if you have a box or something like this and you want to color it, one of the color options or fill options is gradient. And when you choose that, you can choose a variety of different ways to show how the color is distributed in the box. Here on this box, the gradient shows a high concentration of color at the bottom and a low concentration of color at the top. So there's a difference in the amount of color. That's the gradient. So diffusion always goes from where there's a lot of it to where there's a little of it, or in other words, spreading out. So in this container, I've put some breadcrumbs on one side and coffee on the other. Right now, there's a gradient between them. Coffee is much more concentrated on this side, and the breadcrumbs are much more concentrated on that side. If I cover it up and I shake it, simulating Brownian motion because it's moving randomly, eventually, when I'm done shaking it, the coffee and the breadcrumbs are evenly dispersed. And if I were to shake this for another 10 minutes, I'd come back and it would still look pretty much like this. They still would have been moving, but there's no longer a gradient between the two things.
This animation shows an example of diffusion, which is a drop of food coloring in a solution. It also shows how Brownian motion causes diffusion. So, a drop of food coloring is added to water.
As it's added to water, those food coloring particles start off real concentrated, but as they bounce into the water molecules, it spreads out over time. You'll notice when I do it again, the food coloring, it doesn't continue to get darker and darker and darker all over the place, but rather the amount of food coloring that's there spreads out until the coloring is over the entire space of the water. Also notice that once it's evenly dispersed, once there's no gradient, there's no more motion. Let's add that to the diagram in your notes. So let's transfer that into your notes, and you can, I have like color spludges, but you can put little dots if you want. So when we're talking about it in terms of particles or molecules instead of just smudges of color, what we're seeing is that the concentration of molecules, like represented by dots here, to start with is more concentrated in one section, and then it spreads out evenly eventually. It's important to remember, though, that once it's done and evenly spread out, that there are still continuous movement of molecules. And so there's still that vibration. There's just no longer a net movement. Just like if in my example with the breadcrumbs and the coffee, it'd still be moving around and there would just not be any great change in how it looked. You have a spot in your notes to show a gradient. When there's no longer a gradient and it's evenly dispersed, like it is here, we call that an equilibrium, and that's when it continues to move, but it no longer has a difference in concentration. Now that you know how diffusion works, let's talk about how it applies to cell transport. So remember that some things can get in and out of the cell as if there was no membrane at all. And so things that can do that move through basic diffusion, simple diffusion, nothing special, what we've just described, going from high to low concentration. And the things that can do that are the things that are small enough to get through the membrane on their own. So they are small molecules, mostly oxygen and carbon dioxide, but a few others, and they have no charge, so they're not going to be repelled by the inside of the hydrophobic membrane, and so they can just diffuse in and out of the membrane as if there was no membrane at all. Let's take a minute and review. If we take a look, how is this different from that? You'll notice that we have diffusion. Materials are moving in this diagram from the top side of the membrane to the bottom. And so they are moving from where there's a lot of it to where there's less. So that's still from high to low. That's diffusion. But what you hopefully thought to yourself when I asked you how this picture is different is that these molecules aren't going across the membrane like oxygen was in the previous slide. Instead, they're going through a protein. So in my last video, when I talked about the different kinds of proteins, I said that proteins some proteins serve as the doorways for things that can't fit through the membrane on their own. Well, these would be that. Things that can't get through the membrane have to go through a protein, and we call that facilitated diffusion. To facilitate something simply means to make it easier or to help, to make something happen. So facilitated diffusion is when materials need help diffusing. In this case, it's through a protein. So it's exactly the same thing, except it's diffusion through a protein. Just like plain old simple diffusion, it's still passive transport. It still needs no energy. It's just they're going through a door rather than just going through as if there was no boundary at all. This kind of diffusion would be when things need to move from high to low concentration, but they're either too big to get between the phospholipids on their own, like that would be like glucose, for example, or they have a charge. So they may not be big, but they're going to be repelled by the hydrophobic middle. So for example, that would be like sodium and chloride, ions. Let's take a minute and review. The last type of passive transport is, again, a type of diffusion, and specifically a type of facilitated diffusion called osmosis. It's the facilitated diffusion of water across a semipermeable membrane. So it's facilitated diffusion, which means it's going through a protein, and that protein is a special protein called aquaporin. And again, it's a form of passive transport. The cell needs no energy to do this, and it's moving from high to low concentration. I'm about to use the word solute a lot, and just to remind you from our last unit that a solute is a substance that's dissolved in a solution. So in this case, it would be something that's dissolved in water. So if we take a look at the diagram on the side, the concentration of the solutes, those are the things in red, are different on either side of the membrane, as are the concentrations of water. So if we look at the top, the solute is very, very low concentrated, where the water is very, very high concentrated. Conversely, on the bottom, there's a lot more solute based on how much water there is. So the water is in low concentration below and in high concentration above. It's facilitated diffusion, which always means going from down the concentration gradient from high to low. So water is going to be the thing that moves, and water moves from where there's a lot of it, in the diagram it'd be on the top, to where there's less of it. And in doing so, eventually, it moves and balances out the concentration of the solute by having the water move. Kind of like if you are mixing lemonade and you put too much sugar in it. If it tastes too sweet, you can add more water to dilute how much sugar there is. It's basically a similar idea. So again, it's water moving across the membrane, and it's water that's moving from high to low concentration of water. So why is it that's water that's moving and not the solute? Well, if we take a look at this diagram, the water, which in this, the water, which again is shown in blue and the solutes are in red, the water can fit through the permeable membrane, and the other material, in this case it's sugar, cannot. And so because the other material can't move across the membrane and the water can, water diffuses to balance out the concentration. So we see that some of the water molecules will move over here, and as they move across the membrane to the left, the amount of water will go down on this side, and this actually goes higher water here. And so I'm not going to draw it with all the little dots because that would take a long time, but there would be far more dots on this side compared to how much solute there is, so that by the time there's no more net diffusion, in other words, by the time there's an equilibrium, the solutes have an equal concentration because the water has balanced everything out. So a few seconds ago when I described osmosis, I mentioned it's a type of facilitated diffusion that goes through a protein. That protein is named aquaporin. If you're familiar with Spanish, agua means water, and then pores like a hole. So aquaporin is a specialized protein that is specific to water. Remember, water is one of those molecules that's small enough that it can get across the membrane on its own just through simple diffusion. But there are times when the cell needs it to go across the membrane much faster. In other words, it needs it to get water in very, very quickly. There are many examples, but one that you might think is cool is that if you've ever heard of a Venus flytrap, that's the type of plant that can capture a fly by closing real quick. Plants don't have muscles. The way that the Venus flytrap closes is it actually changes how much water is in certain cells very, very quickly, enough to trap a fly. And so in order to move that much water, because water has difficulty getting through this hydrophobic center, it can go through a specialized pore. Now, because water is a polar molecule and it's got charge size, there's actually charges within the aquaporin that help pull the water in and attract to different parts. And so the water molecules it travels through actually kind of flips over as it attracts to the different parts of the molecule. So aquaporin is the channel for the water. And again, it's specific to water because of those charged regions. When it comes to osmosis, there's 3 conditions we need to be familiar with. And so we have specific names for those conditions. So if we take a look at this diagram, we see the little Mickey Mouse shaped creatures. That's supposed to be water. The big hexagons are meant to be solute like sugar. In this case, even though there are probably, if I count it up, the Mickey Mouse shapes, there's probably at least as many or equal or more water molecules on the outside. The concentration, in other words, how much sugar there is compared to water, is much greater on the outside than on the inside. We call this type of solution a hypertonic solution. These have solutes that are greater, the solute concentration is greater outside the cell, and the water concentration is greater inside the cell. To balance out the concentration, water will move from where there's a lot of it, inside the cell, to where there's less of it, outside the cell. So because of that, because the cell is losing materials, it's losing mass, it's getting smaller, this cell tends to shrivel up. In a hypertonic solution, the cells will get all shrivelly. In a plant, plants have cell walls, so they can't shrivel the same way an animal cell will, but the cell membrane will actually pull away from the cell wall. On this diagram, take a second and add in some arrows to show the direction of water movement. Add in a couple. I would say maybe at least 4. But as you add in the arrows, make note to make at least 1 arrow or 2 really little ones inward, because it's important to note that water is moving. It's Brownian motion that's causing it, so it's moving in both directions. It's just that there's more water that ends up moving out than in. My memory trick for this isn't great, but if you think about having a lot of sugar, sometimes people who eat a lot of sugar get a sugar rush, and we call it that they're being hyper. And so my memory trick is if you have a lot of sugar, a lot of solutes, then you're going to be hyper. Okay. So solutions with more solutes are hypertonic. By the way, hyper means above. So the solute concentration is above. So if we look at the next kind of cell, we have hypotonic. Hypo means under. So in this case, the solute concentration outside is under. So the solute concentration is low on the outside, meaning water concentration is therefore higher on the outside. So water moves from where there's a lot of water to where there's less. So from high to low concentration, meaning that water will move into the cell. So label that in your table, and then draw a bunch of arrows with some big arrows, at least 3 or 4, some big arrows going in, and maybe 2 smaller ones going out, because again, Brownian motion means that water is always moving randomly. It's just by the end, more water is going to move out or move in than out. In this situation, the cell is going to gain mass because more water is going to move in than moves out, so the cell is going to get bigger. In hypotonic solutions, the cells gain so much mass that in an animal cell, the animal cell may gain so much water that it can burst. It doesn't have a cell wall, so its membrane can only expand so far. We say it lyses. Conversely, a plant cell has a cell wall. It helps it remain rigid, and so it's supported by that, and it doesn't burst. It just plumps up really rigid, and we call it turgor pressure. Again, I have a memory trick for it. Again, it's not a fantastic memory trick, but I always think of in that hypo has an O in the middle of it. So I think that O gets like a big plumped up fat letter. So hypo, the cell is going to get plump and fat. Finally, we have one more condition, and that is the ideal condition. Remember, our bodies always want to maintain homeostasis. And so in this situation, the concentration of water inside the cell and outside the cell are the same. And we call this type of solution an isotonic solution. And they have equal concentrations. So consequently, water moves both into the cell and out of the cell, because remember, there's always movement. Water moves both into the cell and out of the cell in equal amounts. And consequently, the cell size is stable, which is, of course, what we want, because we don't want our cells to burst and we don't want them to shrivel up. So on your diagram, add an equal number of arrows that are relatively equally sized in both directions. And of course, fill in the table. So let's take a second and review osmosis. Now that we've talked about passive transport, let's talk about active transport. Some teachers in books talk about it differently, but I say there's 2 major ways. If we look at this first way of major method of active transport, it may look a lot like facilitated diffusion, but there are 2 very important differences. If we look and see how this is different, one way that hopefully you noticed is that there is an input of energy. There's this little burst there showing that the cell is doing something, inputting energy to make it happen. And if you were really astute and observant, you may have noticed that the concentrations are different. Remember, in passive transport, material always moves from where there's a lot of it, from high concentration to low concentration where there's less of it. But here, the net movement is this arrow is going down into, I assume, what's supposed to be into the cell. And so consequently, if we take a look, it's going from where there's less on top to where there's already a lot. So to get into your notes, active transport is moving materials across the membrane that goes against the concentration gradient. So from low to high instead of high to low. It's a little bit like swimming upstream. If you're swimming with the current, sometimes you don't have to swim there. You can just sit and let the current carry you. That's the whole point of a lazy river at a water park. If you're going to go against the current, you have to work and move faster, move harder, and put more energy to go in the opposite direction. It's a little bit like that. So there are, like I said, in my book, there's 2 major types. The pictures I've been showing you so far are examples of protein pumps. So those are transport proteins that instead of just being a channel or a way for the material to move through the membrane that maybe is too big or charged, it actually is doing something. It's a little bit like an elevator. The material gets in on one side, and then the elevator needs power to move up to go to the next level. And so these require, usually there's some kind of a door shape or a change in shape to force the material across the membrane. A really good example is something called a sodium-potassium pump. It's really important in cell communication and sending messages. The other kind is called cytosis. It is bulk movement. So it's a way of getting very, very large molecules in, or a whole bunch of molecules all at once. And that because they're molecules that are either proteins themselves, and so they can't fit a protein through a protein usually, or they're just really, really big molecules. They can't fit through a protein, so the cell actually has to change shape to do it. It does so using something called a vesicle. If you haven't gotten there in your notes yet, a vesicle is basically like a little shipping box that the cell uses, except instead of a box, it's a little membrane sac, and we'll cover that in a sec. Here's an example one called a proton pump, which is moving hydrogen ions across the membrane. Here's the famous sodium-potassium pump that I mentioned. It works in a similar fashion, a little bit more complex, but a similar fashion. In this animation, we can see an example of a protein pump. The material that's moving, the orange or brown or whatever color balls there are going from high to low. And every time a material moves, the protein changes shape, and that requires ATP. ATP is the molecule that supplies energy. Cytosis, again, is when we're moving bulk material. So in this picture, it shows a whole bunch of stuff, but again, it could just be a very large molecule. And so we have basically the picture is moving in two different directions. The top part and the bottom part are basically the same. So the first one, let's talk about that, is endocytosis. Endo meaning in. So endocytosis is when material moves into the cell. So what you'll notice is this double line here is meant to be membrane. So the membrane makes a pocket. The material that's going to move in collects in the pocket, and the membrane pinches off so that once it's fully pinched off and completely closed, it can separate from the membrane. In doing so, it avoids like poking a hole in the membrane and having everything leak out. And so once it pinches off, then it can open, it can be inside the cell, and then that material is wrapped in what was membrane, and so it's a membrane-bound organelle. Once the material is enclosed in a vesicle, then the vesicle can do what it needs to do, whether it's destroy the contents because it needed to break it down, or use those contents in some other part of the cell. Endocytosis has different words for whether it's bringing solids into the cell or liquids into the cell. We call it phagocytosis or pinocytosis. My memory trick is phago starts with a f sound like food does, so sometimes it's called cellular eating for food. This is how a single-celled organism like an amoeba would eat. It would take in a smaller organism like a bacteria cell. Pinocytosis is when the material taken in is a liquid. It's sometimes referred to as cellular drinking. You don't really need to know those terms. It's really more of an FYI. Finally, the last type of cytosis is exocytosis. Exo meaning exit, material is leaving the cell. In this case, this vesicle came from someplace inside the cell. Maybe the cell is making proteins, like your pituitary gland is making hormones that need to be used elsewhere in your body. And so those proteins need to be transported out of your cell so they can be carried somewhere else. And so consequently, this vesicle maybe came from some rough endoplasmic reticulum or from the Golgi apparatus that's packaged it up. And now that it's contained, what it needs to get out is contained in a vesicle. That vesicle has membranes. And so that membrane can fuse with the cell membrane. And once it fuses with the cell membrane, it will ensure that nothing leaks out and it can release the contents to the other side without the cell leaking. So we call this exocytosis. It's the removal of material out of the cell, either intentionally because they're a product of the cell or it's waste the cell needs to get rid of. And it involves a vesicle enclosing that material, fusing with the membrane, and opening up on the other side. This would be, again, how any of your cells make a product that's leaving your cells. When you cry, this is what happens. And again, memory trick is just use root words, exo, exit, things leave the cell. Of the cell producing a protein. It does so on the rough endoplasmic reticulum. That is wrapped in a vesicle that carries it to the Golgi apparatus. Both structures are made of membrane, so it can fuse with the Golgi where it can package it up, make some modifications. And then after it does that, the Golgi repackages it here into another vesicle, which fuses with the membrane and opens up on the other side. Let's take a minute and review. That's it for this video. Let me know if you have any questions.